now our second speaker for the Conservation and Restoration Research theme, Amy MacDonald. Understanding the distribution of generic variation in Ramarama, as Colin mentioned, and its potential implications for conservation. Amy has recently completed her Master of Science at Massey University in Manawatu. Her field of study is plant genetics and conservation, and her master's research focuses on the population genetics of the threatened native species, Ramarama. Tēnā kamahinga koe Amy, wiki mai, kia ora.
I'll be discussing my master's research, which was very kindly funded by MacDonald and involves the investigation into genetic variation within Ramarama. So I'll first begin by providing a bit of information or a bit of background both on Ramarama and on what I mean by genetic variation. Then I'll talk a bit about my research, both in terms of what I did and what I found out. And then lastly, I'll discuss how what we learn can be applied to Ramarama's conservation. So, Ramarama, or Lophomus blada, is as we know, a plant species that is endemic to New Zealand. It is a shrub or small tree species whose leaves are predominantly green in colour and have occasional red pigmentation and characteristically bubble-like appearance. The species is distributed throughout the North Island and the northeastern corner of the South Island and is generally found in lowland and coastal forests. Where the species co-occurs with closely related species Lophomus or Purata, also known as Rahutu, the two species are able to produce fertile hybrid offspring. Both species are members of the myrtle family, which means they are vulnerable to the pathogenic fungus known as myrtle rust. Ramarama in particular has been identified to be highly susceptible to the disease, and very little resistance has been observed in natural populations. This means there's high potential for the localised extinction of the species. One way we can help contribute to its successful conservation would be to have a better understanding of the amount of genetic diversity present within the species. This is based on the theory that the more genetic diversity a species has, the better able it is to adapt to any new environmental changes. Therefore, maintaining a species' genetic diversity is required to preserve its adaptive potential. The way we investigated genetic diversity was by assessing variation at a population level. A population just being a group of individuals at the same location. We would determine the genetic composition of one population and then compared how genetically similar or genetically different it was to another. And this is simply just dependent on the amount of genetic material, or in the case of plants, the amount of seed and pollen that's been exchanged between them. This method also allows us to determine how genetic variation is distributed across the landscape and identify any potential patterns that may emerge. So with this information in mind, the questions our research hoped to answer included how much genetic variation is present within populations of Ramarama, how is that variation distributed across the landscape, and what is the genetic structure of the species. So to answer these questions, we first collected leaves from 20 to 25 individual plants of Ramarama for each of the 18 populations identified. We then collected additional leaves from 20 individuals of Ramarama and Rahutu hybrids from two different populations. And this was just to assess the potential effects of hybridisation on the genetic diversity of the species. Lastly, we collected from 12 individuals of Rahutu from one South Island population. Local iwi and hapu were contacted for permission, and we even had some iwi representatives come along for some of the collections. Some samples were then analysed using microsatellite markers, which are simply just tools that allows us to measure the genetic diversity and structure of the species. So for a little bit of context, the value we use to measure genetic diversity is a range of 0 to 1 meaning high genetic diversity, and 0 meaning low genetic diversity. And we found that for populations of Ramarama, the amount of genetic diversity ranged from 0.36 to 0.57, placing Ramarama in the mid to low area of our genetic diversity range. When we look closer at how this genetic diversity was distributed, we found that populations in the upper North Island contained high genetic diversity, while populations in the lower North Island contained less genetic diversity. And when this relationship was tested, it shows there is a significant linear decline in genetic diversity as you move from north to south. One possible explanation for this pattern is the effects of glacial cycling during the Pleistocene, which heavily restricted forests and therefore Ramarama to the upper regions of the North Island. As global temperatures warmed and glaciers retreated, there could have been a serial expansion of Ramarama further south, with each new population being colonised by a population that was both smaller in size and contained less genetic diversity. Potentially contributing to the north to south patterns we see within populations today. In addition to this north to south pattern, our genetic structure analysis also identified the presence of 5 genetic groups within populations of Ramarama. These 5 genetic groups roughly corresponded to the population's regional location as indicated in the figure. Interestingly, the boundaries of these genetic groups also roughly corresponded to known biogeographical patterns present in other New Zealand flora and fauna. Majority of the populations were found to share a lot of genetic variation with each other, suggesting that there's high amounts of seed and pollen being exchanged between them, particularly between populations that were geographically close. Before I talk about how this can be applied to Ramarama's conservation, I'd just like to briefly talk about hybridisation between Ramarama and Rakutu. So our 2 hybrid populations were found to be a genetic mixture of both the species, but they're also found to be very genetically different from each other, as well as containing a lot of high amount of genetic diversity, about the same level as our highest populations for Ramarama. This is partly expected, as when species hybridize, you're essentially taking genetic diversity present in one species and the genetic diversity present in another, and combining it into a new population, potentially creating a new source of genetic diversity, as well as new combinations of traits. If these hybrids can then be backcrossed or crossed back into the species' parent populations, these traits can potentially be shared between them. Therefore, it's possible that hybridisation between Ramarama and Rakutu could allow the introduction of new genetic variation into both of the species, as well as potentially now sharing of traits that could be beneficial for possible numerous resistance or even climate change adaptation, though this is dependent on the hybridisation mechanics of the species. In terms of Ramarama's conservation, the results discussed here help to aid seed bank collections by ensuring they represent the entire genetic range of the species, as well as help inform future ecological restoration activities, such as the translocation of individuals between populations to boost diversity. Additionally, understanding how genetic diversity is distributed across the landscape can be used in future research into possible minerals resistance or climate change adaptation within the Ramarama. Finally, this results acts as a snapshot of the current genetic structure of the species, which we can use to track any changes in the genetic diversity caused by the effects of wheel rust. All these efforts are more can help with the preservation of this tiny species for future generations. Kia ora and thank you for listening.